My name is Alexis Nolin-Lapalme, and I will be your instructor during that series of tutorials regarding structural biology and structural analysis of proteins, but also of DNA, as both can be analyzed through the same methods. So we'll go over multiple analysis pathways, ways to analyze both in terms of tools, both in terms of imaging, and both in terms of tweaking the structure to make either great pictures or great analyses that's actually allow you to funnel these tools or this knowledge into different pathways of both drug discovery, but also of different areas of scientific research. So today, we're not going to go over any analysis per se, but more over an overview of why to use such techniques, what type of tools we'll be using, and what type of mindset or what type of ideas you must have when you start a structural analysis design or a design experiment. So let's hop on the wonders of the internet and describe the various type of pictures or different types of analyses and where the protein structural analysis is being used in research. So simply writing protein structure into your favorite web browser, we can see there's a vast array of different types of pictures. It's actually used in posters. It's actually used in scientific websites. It's actually frequently used into research. That's actually highly picture that's going to be used with one of our software. That's the kind of picture we're going to be able to produce. These are actually produced with much. This looks. Yes, so this could be also something that looks like it's being taken by the software we're going to be using. There's high-end software. Each software makes protein pictures slightly different. However, they use the same principle, the same illustration. There's usually ribbon. Here we can have the structures of the estimated surface. You can have weirder ones. You can have this kind of highly filled representation. Sometimes you can have both overlapping together. And these are used in a vast array. Sometimes you have pure scientific research. For example, this one that was researching HSPs. That was basically looking at key structural parts. Sometimes it's part of a bigger scale. Sometimes you're looking at, for example, like right here, what it looks like. It's actually looking at ligands, so different amino acid binding NAD plus in this case, the way that it interacts, it moves, the different moieties that would possibly allow it for interaction with maybe an inhibitor. So it's not only part of drug discovery, but it's part of pure research, basic research. But it can be used in a vast array of subjects, and therefore it's a key tool that's going to make you a much stronger candidate in laboratory positions, as it's a highly versatile knowledge base that can be applied in different scientific questions. So then, now that you know a little bit what it looks like, where it's being used, what's important, what kind of software, actually more importantly, free software, as StudentPharma wishes to allow knowledge to be as free as possible and to allow you to have as much open source content as possible. So there's a lot of tools. Some of them are expensive, some of them are free. We'll go over some of the main ones we're actually going to cover through these series. So there's two main free software that are being used in laboratories. The first one is called Pymol, which is probably the most well known. So this is what the interface looks like, but we'll go more into the website. So it's actually offered by the Schrödinger company that offers great tools ranging from pure docking, we'll go over that a little bit later, to protein imaging. They actually have two software. They have Pymol and they have Maestro. Maestro is the high-end tier of their protein imaging software, which can actually be obtained freely as well for academic email addresses. So, for example, I'm a McGill student. I have a McGill email address. Using a McGill ID can allow me to get e-software for free, as it's automatically recognized as an academic user. So this is Pymol. It's offered through the main OSs, but will not be the one that's going to be used today or in this series. The reason is that it's highly covered in multiple webinars. So we'll do another approach, which is actually using what is called UCSF Chimera. So UCSF Chimera is also free, but the comparison would be Pymol would be kind of the Linux operating system, while Chimera would be the Windows or the Mac. They both do pretty much the same thing, slightly differently. There's advantages for each one of them, but Windows and Mac can be used easily without any knowledge of programming, of the Unix command line, Bash. It can be simply used through clicking through menus and getting results right away. So this is what we're going to use today for simply ease of use. Pymol is a very, very powerful tool as well. It's actually very powerful also because there's multiple downloadable content that allows to increase the power of the tool. However, we want to keep these as simply as possible and also to explore UCSF Chimera a little bit more. So here's the website. You can explore it in different ways. They actually came out with Chimera X. Will not be covered today, as personally I am more used to the original UCSF Chimera, but do not hesitate to hop on UCSF Chimera X, as it offers different tools and possibly better pictures as well. So here on the website, there's a documentation where I invite you to read through. There's a lot of stuff, but what's interesting is they actually have tutorials. And the tutorials regard content that will not be covered in detail in this series. So it allows you to actually increase your knowledge base regarding the content. It's actually written in Python, actually most of it, not everything. So it allows you, if you're familiar with Python, you can actually play around a little bit more with the software. So again, there's some videos, there's some release. If you want to hop into download, choose, if you know your OS, simply download the according version. Remember, there's a big difference between 32, there's a difference, not a huge difference. Well, it's going to make a difference between 32-bit and 64-bit as operating system, and simply download. So you can see they're actually quite recent. So right now we're in June 2018. Therefore, 2018 is quite recent. So we can see that in my case, my laptop is a Mac OS 64-bit. Therefore, it's a quite recent patch. Last time was bit. So you can download it here. I will not download it again, as I already have it on my computer. So just hop on it, explore. So once it's downloaded, it should be an easy wizard. So you just click, read through the website, and once it appears on your computer, you just drag it through apps, and it should appear as this icon. Maybe if I put it in a slightly more colorful, perfect. So it's basically a blue circle with a black half circle inside. Once you open it, the layout's going to be slightly different between you and me. So basic Chimera is free. However, it doesn't ask your information until 27 days. So I reinstalled it recently, and it's basically prompting me with 27 days. I'll do it later, but just know that it's entirely free. It's never going to ask you for money. Okay? So if you've used it before, or you're using it today and will use it again, you'll see your previous structures right here. So, so here I have multiple structures right here. However, it should look like either this or completely black. Okay. So if I'm loading a structure, if I remember, well, this is yellow fluorescent protein, I'll simply erase it so it looks like your the interface you have right now. Okay, so don't worry about the commands I've done just now. We'll just go over it rapidly over the commands. So UCSF Chimera. Here you have the interface. So basically, when you have a protein, it's going to show right here. Here you have your error line. Okay. Here you have your files. So under file, you can open or fetch by ID. What does that mean? Well, proteins for computers cannot be illustrated as ribbons or other highly complex structures. If we hop on internet, they're actually written as PDBs. That stands for Protein Data Bank. So it's simply a file type that looks like this. So here you have atoms. You have the atom number, the element. So here you would have carbon. You have carbon, oxygen, nitrogen. And of course, because there's multiple atoms in the single amino acid, you have the amino acid they're related to. Then you have the chain name, the sequence number. These don't really matter a lot, but just remember that sometimes proteins are as dimers and they crystallize as dimers. And then the coordinates. So basically, a PDB is a set of coordinates for atoms that are classified through their amino acid and their sequence. So basically what a protein imager does, it's basically fits these coordinates and associates them with atoms. So usually the software knows what is the radii, what is the usual properties of these atoms. So basically what the PDB says is basically, okay, so here we're going to have a nitrogen, it's going to be part of an aspartame. So it's going to build that kind of framework of an aspartame. Let's go. ASP. Okay. So it's going to build that framework, but not only the framework, but it's going to tell the imager that this is actually being related to this particular amino acid. So the software is going to identify this as, let's say, aspartame number 21. Okay. Okay, so now that's clear. Let's see where we get these, right? Because they won't follow this guide. You won't make them personal. What you'll actually do is go on the PDB, which is funny because it's the same name as the file, but Protein Data Bank is actually the website where people actually submit their structures. So here we can see latest entries. So these have been recently submitted. As to be published, you need to submit your crystal structure. So when a lab makes a new, oh, that's pretty interesting. Okay, so this is actually highly complex, and you can see there's actually RNA, of course, because this is a ribosome. There's RNA and there's protein mixed together. So actually PDBs can fit also DNA because it's simply atoms, and there's some basically amino acids, as you know, have the same kind of atoms as DNA. They're just arranged in different molecules. So you can have this really highly complex structures. So actually, let's just do a simple one, right? Just an easy one, insulin. Really very simple peptide. Okay. So what you presented here is the paper that you authored. As you know, insulin is a quite old molecule. We've known about it for a long time. So what information is given to you? Well, the name, the ligand. So here we have zinc and chlorine. Sometimes there's going to be very specific ligands. If you have a drug receptor, you're going to have a more complex name for the ligand itself. The residue count, so how many amino acid is in the structure. The resolution. What is the resolution? Let's hop again on our trusty web interface, resolution, protein structure. So the that's a great picture actually. Thank you to this the people that posted this. Okay, so what do you see here? Well, when you create a protein structure, you start from something, not exactly this, but you start from a crystal of a protein. Do I have? Oh, that could work actually there. And you get you just refract electromagnetic waves to disturb the atoms, and you can actually measure it. But then the precision of your software or your readings or your experiment, actually it's a mix of everything, is actually going to allow you to get different precision of the estimation of where the amino acid sits, right? Because you get that cloud of electrons. And then you can say, well, something is in here. What is it? Well, the higher your resolution, the closer you can estimate actually the position of every atom, right? If not, it's going to be here, it's just an average. As you can see here, it's actually cut, but there's actually something here, but the resolution is so bad that you cannot actually estimate it. Actually, it looks like this, right? Because it's highly complex, and you need to determine where is your amino acid. And this becomes highly, highly relevant in drug discovery, because maybe that pocket right here is too small or too big for a drug, and right here it's actually fitting, but you will never know it if the resolution is too low. So that's what the resolution means. It's basically the quality of the related structure. How precisely does it fit the actual position of the amino acid? And this is the method. So X-ray diffraction, so they just use X-ray. There's cryo-EM also as a method. So this is the release date. If we click on it, short note, PDBs are always represented through a 4-character code. Okay. So here we have the organism, scrofa. It's a hormone, as you know. Authors, the percentile rank, and some information regarding the protein. So you can have the ligands. Here you can have the molecular structure when it's better. So the class score is how much the score of the amino acid that actually clash with each other, that actually colliding in the structure together. And these Ramachandran and sidechain is just different metrics regarding the quality of the structure. We won't go into many details. This is some extra info regarding the protein. You can have blah blah blah. And UniProt is a great database regarding all information regarding proteins, but we're just interested in the structure. So there's different things you can do. If you want to keep it on in your computer, you can download the FASTA sequence. FASTA sequence will be part of different series, but FASTA sequence are rapidly the amino acid sequence without any structural component, just the series of the single letter code. What we're interested in is either the compressed or the full file. There's different versions, but you can actually have it with the XML. But usually you just download it through the compressed. If you're having a small space on your computer, you don't want to expand too much, it's going to be a little bit smaller, or the PDB format. If you don't want to keep it on your computer, you're just interested in a quick analysis, what I can do, and the beauty of structural software is actually to copy paste it, or just remember it if you have a good memory. And in Chimera, remember we had open and fetch, just fetch by ID. Here you have different databases, UniProt, as we talked before, but there's other ones, but the main ones, of course, because it's automatically selected, is PDB. Just copy paste it here, just fetch. There you go. You have exactly the same structure. Okay. These big things, of course, are atoms, and these are basically the amino acid connecting to, I think this is going to be zinc, this is going to be calcium, chloride. Okay. And as you know, ions are quite necessary to achieve structural integrity of multiple protomers. So here you have an interesting thing. As you can see, it's a dimer. Okay. So this is a quick overview of how to download a protein picture on UCSF Chimera and the different reason why structural analysis can be quite important. In the next tutorial, we'll go over the different menus and the different tools that are within these menus that allow you to do multiple things, from analysis to transformation to movies to animation and to calculation of distance, and actually to edit the molecule and actually docking all throughout these menus. Okay, so regarding that, just stay tuned for the next tutorial. My name is Alexis Nolin-Lapalme, and see you later.